Namaste students. Today I am going to discuss about the cell lines. What are the cell lines? We have cultured the cells in the primary cell culture and the secondary cell culture. In the primary cell culture, we culture cells from the tissues, fresh cells. Okay, the fresh cells which are cultured are known as the primary cell cultures. And when we culture these primary cell cultures into another media and subculture them, then they are secondary cell culture. They are secondary cell cultures. And then these secondary cell cultures will make some layers and they will grow and they will divide on each other. They will create the cell lines. So these are the cell lines which are the dividing cells of particular kind of cells. So these are the cell lines. Okay, on the basis of the lifespan we can categorize these cell lines into two types. The first one is finite cell lines. So as you know that in the primary cell culture, the growth of some cells is limited, means they will uptake the media for a limited period of time, and when the media is depleted they will stop dividing. Okay, they do not have capability to divide more, and they can divide for only a limited period of times. So the cell lines which have the capability to divide for a limited period of time are known as the finite cell lines. They will not divide continuously, but they will divide for a limited period of times. And when they will divide for a limited period of times, these cells can go to the death. Okay, they can go to the death. They will not proliferate, they will not divide. So what will happen? They will go to the death. The death is known as the senescence, cell death. Okay, and most of these cells are derived from the primary cell culture. Okay, these finite cell lines are derived from the primary cell culture. Okay, it is clear now. The continuous cell lines. What are the continuous cell lines? Continuous cell lines are different from the finite cell lines. As we know that these cells can grow, these finite cells can grow for a limited, and they can divide for a limited period of times. These continuous cell lines are different. These continuous cell lines have ability to divide indefinitely. Okay, they will these continuous cell lines will divide indefinitely, so there is no any limitation. Okay, and these cells are originated from the finite cell lines. They are originated from the finite cell lines. They are not prepared by themselves. No, they are derived from finite cell lines. Okay, when these finite cell lines will get transformed, they will develop into the continuous cell lines, and these continuous cell lines will have capability to divide indefinitely. They will divide continuously, and this transformation can be called as the mutation in the finite cell lines, which will develop into the continuous cell lines. These continuous cell lines can be developed by either by the mutation, or they can be synthesized by the chemically, or some induced uh, mutation method. And the cell cultures which are prepared from these continuous cell lines are immemorial, means they will not die until the media is depleted. Okay, They will not die until the media is depleted. So they will not die. And these cells are less adherent. They cannot get adhere onto any surface. Means they will adhere, but they are less adherent and they will grow very fast other than the finite cell lines. Okay, and they have some nutritional requirement also, which will divide when they will find some nutrients from the sources. Okay, so the multiple layers of these continuous cell lines will be used for the animal cell culture. So these are the continuous cell lines. Okay, so the examples of these cell lines are the human cell lines, such as MCF7, HL60, and HELA, human cervical cancer cell lines. What is the cell strain? Okay, that is also a term. Lineage of cells originate from the primary culture is called the strain, cell strain. So the lineages of the cells which are originated from the primary cell culture is known as the strain, and these are derived from a primary culture or a cell line by positive selection or cloning of the cells having specific properties. Okay, these so in summary we can say that in the primary culture some cells are very specific. Okay, some cells are very specific, so we select these cells, and these selected cells can be known as the cell strains. Okay, these lines of the cell lines of primary cultures are known as the cell strain. Okay, so that's all about this lecture. In the next video we'll discuss another lecture. Thank you very much.